So thanks everyone for joining the prediction modeling and machine learning seminars. As I think I said before to Gustavo, I'm in a public space. I hope that there's not too much noise and that you can hear well. So thanks everyone, and particularly to our speaker, Gustavo Sudre. Gustavo is the Rosetrees Pears Chair of Bioinformatics and Professor of Genomic Neuroimaging and Artificial Intelligence at the Institute of Psychiatry, Psychology and Neurosciences here at King's College London. He's also leading the OPM-MEG program at the Pears Maudsley Centre, and it's also co-leading the Precision Psychiatry and AI for NeuroDevelopmental Analysis lab. We are very, very lucky to having him today talking to us. So thanks very much, Gustavo. Let me tell everyone that we will have our talk, then there will be a space for questions and answers, so that Gustavo will be happy to discuss with you about what he's presenting, and also letting you all know that this is going to be recorded. It's actually being recorded right now. If anyone has any problem with it, just let me know. And also that if anyone would like to be added to our mailing list about these seminars, please feel free to leave your email address in the chat, and I will add you to the list. So I think that's everything. So go with it, Gustavo. I leave it to you. Thank you.
All right. Thank you, Raquel. So thank you for the invitation. So when Raquel approached me about giving this talk, I was upfront with her that I haven't been doing much prediction modeling lately, and because most of my work has been on the discovery side. So we agreed that I'll present what I've been up to in discovery and also the plans that I have for returning to prediction, and that's what this talk is. So part of what I'm hoping to get out of this is feedback from everyone here on the design choices that I'm making for this upcoming prediction work. So just a little bit of context on me. I came to King's in late 2024, and the past year or so has been spent growing the lab, which we call the PANDA lab. So the Precision Psychiatry and AI for Neurodevelopmental Analysis. And we also thought that PANDA was going to give us some options for cute logos, right? So, we've been just putting in place the legal, computational, and collaborative infrastructure that we're going to need. So a lot of what I'm going to describe today is in the form of plans and design, not necessarily finished results. So another thing I want to flag from the start, I am not a clinician. So if questions come up, they're about the clinical practice, I'm just going to say, I don't know. I have no problem saying that. So today I'm going to lay out the prediction program of the PANDA lab. So it's organized around 3 clinical problems that we see in child psychiatry. So who is at risk? How symptoms unfold, and which treatment will help. And I'm going to be quite transparent about what is built, what's in design, and where I most want your input. And then I'm going to finish this talk with a couple of slides on the Pears Maudsley Centre, which is a new facility that we are building precisely because the data problems that I am about to describe, they will not be solved just by better models alone. So let's fast forward. See, this was working before. Perfect. Okay. So let me give you some numbers. About 1 in 5 children worldwide face a serious mental health challenge before they turn 18. And this isn't really news to anyone, but it sets the scale we're building the models for, and it sets up why getting prediction right matters. Now, I know this audience that does prediction modeling in medicine broadly. Some of you work in cardiology, oncology, genomics, but I know that every one of those settings has its own hardships. And child psychiatry has a particular set that I want to be clear about them because they're going to shape the design choices in everything else that's going to follow. So first challenge here is that the samples are small relative to the heterogeneity. So a few hundred to a few thousand is a typical clinical cohort against a phenotype space that's enormous. Then second, the developmental change is confounded with disorder change. Say a 9-year-old's brain is fundamentally different from a 14-year-old's brain, and we're trying to predict outcomes across that window. So most of our predictors and outcomes, they co-evolve. And then third, comorbidity is the rule. It's not the exception. So the diagnostic categories that we work in, they were built for clinical convenience, and the biology doesn't really respect them. We always say that the child doesn't read the diagnostic manual. And then fourth, and this is the one that I'll come back to repeatedly today, our cohorts, they systematically exclude the children who most need the help. So severe symptoms, they mean that there's more movement in the scanner, lower IQ, more behavioral disruption, and the standard quality control pipeline filters them out. And you add to that the well-documented demographic underrepresentation in most pediatric neuroimaging samples, and you have models that may generalize to the easier patients, but fail in the harder ones. Now, keep these four problems in mind, and they will come back when I talk about validation, and again, then when I talk about the Pears Maudsley Centre. Okay, so these are the three issues that we're going to be solving. So first, of course, they are not exhaustive in clinical psychiatry, but they cover most of what a clinician actually wants to know when a child walks into the office. So the first one is risk. So who will develop the disorder? For us, this is going to be anchored in our recently funded Welcome project on OCD, Obsessive Compulsive Disorder. And that's predicting the onset risk before the symptoms crystallize. The second issue is trajectory. So once a child has the disorder, what happens next? So this is where I have some published results on a predictive model from 2021 and a clear plan to replicate and extend that. And then the third is treatment response. So of all the available interventions, which one is most likely to help this child? So this is where we also have some published work using supervised models, and we're now transitioning to a more unsupervised approach. Now, running underneath all these three issues is a discovery pipeline that I will turn to in the last session of the talk. So the discovery work is not predictive in itself, right? But it tells us which features have any chance of being useful predictors, which is a precondition for building the next generation of models. Okay, so let's go for the first problem, predicting OCD onset risk. So you might know OCD typically takes years between when the symptoms first appear and when a child gets a clinical diagnosis. Now, some of that delay is help-seeking behavior. Some of it is symptom recognition, some is service capacity, but the consequence is always the same. By the time that most children present, symptoms are already entrenched, and the families have already developed accommodations, and the natural course is harder to change. Now, if we could identify children at elevated risk while they are still subclinical, there is a reasonable evidence from the broader anxiety disorders literature that early intervention can change trajectories. But that's a big if. It depends on whether the prediction at that stage is actually feasible. So this is the question that our project is built around. So the project is called Prediction and Early Intervention in Obsessive Compulsive Disorder. And it funds the work that I'm about to describe, and I'm going to be transparent that this work is just spinning up. There are no results yet, and what I have is a design and a set of methodological choices that I would like some feedback on. So let's go forward. There we go. The scale. So this is not a small n problem, at least not for child psychiatry. So we have access to 9 longitudinal cohorts. They're spanning the 1990s to the 2020s. And these are across 9 high and also middle income countries. Together, that adds up to about 88,000 individuals with about 2,000 OCD cases. Now, the event rate is about 2%. So that's a class imbalance problem, more so than a small sample problem. And then we have 700,000 individuals from the South London and Maudsley NHS Trust, their electronic records for real-world validation. where symptoms of obsessive-compulsive disorder and also OCD diagnosis, they have already been extracted using validated NLP tools. So the cohort design is three-tier, and that's on purpose. They have the discovery set, and that's around 43,000 individuals across three cohorts, where we're going to develop the model using nested leave one cohort out cross-validation, and that's going to give us an unbiased performance estimate within the discovery sample. Then replication is around 44,000 participants across 6 independent cohorts that the model is never going to see during training. So that's the actual external validation step, and it's where most of the psychiatric prediction tools, they usually fail. And then we have the EHR, and that's our third tier, right? That's the real world data with all the messiness that comes with it, and the true deployment test. So the expectation, honestly, here is that the performance is going to degrade from discovery to replication to EHR. And the question is by how much and what that tells us about clinical translatability. Now, one thing that I want to note here is that the cohorts, the 9 different cohorts, they span 30 years across multiple countries. And that means that we're going to have to disentangle true risk from cohort effects. And we're going to do this by including birth year and cohort as explicit covariates, and by testing for cohort by predictor interactions. And if we find them, then we're going to report them instead of just averaging them out. Then there are 2 design choices that I'm putting this on the slide. So first here, there's a dual framing. So we're running this as both a survival problem and also as a binary classification problem. So survival is a primary analysis because the research question is when OCD emerges, not just whether it ever does. The clinical decision that a parent or a clinician needs to make is going to be informed by risk over a defined horizon, not by lifetime probability. So for that, we're going to use Cox proportional hazards as the interpretable baseline, and then random survival forests for nonlinear interactions, and DeepSurv for high-dimensional integration. The binary classification here is secondary, because basically including that for comparability to the prior literature that has mostly used the fixed horizon binary outcome. Of course, we're going to report both, and we'll say which we think is more clinically informative. And then the second thing here in this slide is that the model is built up stepwise instead of just throwing together one blob of features. So there are 4 steps, and each step is evaluated against the previous one as a comparator. So in step 1, we're going to use routinely available data. So obsessive-compulsive symptoms in childhood, family history of OCD, social demographics. Basically, that's the model. Model 1 is the model that a clinician could, in principle, build today, just using a questionnaire and clinical interview. Everything else has to earn its place by improving on this baseline, and that's measured by time-dependent net reclassification index. So what do I mean by that? So in step 2, the second model is going to add genetic risk to those features from model 1. And genetic risk here is multi-PRS, polygenic risk scores, instead of just using the single OCD PRS, because the genomic literature suggests that transdiagnostic genetic risk may carry as much signal as disorder-specific risk. And we're also going to add the rare copy number variant burden that's relevant to OCD. Now, in step 3, we're going to add cognitive and neural features. So cognitive control, we're going to use measures just for the cohorts that have them. But neuroimaging, we're going to have 4 different cohorts that have MRI data for about 35,000 individuals. And we're going to add those, focusing on brain circuits that are implicated in OCD, not just doing a whole brain analysis because that will literally blow up our feature space. And then 4 is when we do stratification and contrast. So if we confirm that our data has a bimodal age of onset pattern, then we're going to stratify and ask which features carry across the lifespan and which ones are specific to childhood versus adolescent or adult onset. And then we're going to run some permutation tests against the models predicting depression and anxiety using the same features, so we can identify which features are truly OCD-specific instead of transdiagnostic features. Now, we find that this stepwise approach here is critical for clinical translation. And why is that? Because routinely available data, if just this model 1 gets you most of the way, then neuroimaging may not be worth the cost of acquisition and deployment. Now, if genetics adds little beyond family history, that's going to inform implementation. So we want to know modality by modality what each is actually contributing. So I won't walk you through the whole validation strategy. I'm happy to talk about that in the Q&A. But like I said, we're going to be comparing the models against this baseline model 1, and we're going to use ProBest and Tripod in reporting everything. Next. So let me just close this OCD section with one slide of why this work package 1 that I just told you about, the prediction work, how it sits inside this broader project, and not just a standalone thing. So like I said, work package 1 is going to build this prediction tool, and that's what I talked about in this first 15 minutes of the lecture. But then work package 2 is going to ask people with lived experience, parents, clinicians, commissioners, across 3 different countries. So we have Brazil, we have Sweden, and we have the UK, asking them how and when and by whom such a tool should be used. And this part really matters because predicting a child's risk of OCD has implications that are well beyond classification accuracy, right? Think about it. So what threshold should trigger the intervention? Or how do we even communicate a probabilistic estimate to a family? What are the harms of telling a parent that their 7-year-old has an elevated risk for a disorder that they may never develop, right? So these are questions that we cannot answer alone, and work package 2 is going to produce a consensus framework that constrains how work package 3 deploys the tool. And that's what work package 3 is doing, right? It's the early intervention pilot. So we have a parent-led digital CBT. That's a program that has already been piloted in Sweden, and it showed meaningful effects on family accommodation. And there are open trials now planned in the UK and Brazil. So the work package 1 tool is what identifies the families that are going to enroll in this intervention. So the prediction 1 model is not academic, it's the gate mechanism for who gets offered an intervention. Now, this integration is the answer to a question that often comes up in psychiatric prediction work. So even if you build like a great model, right? So what then? So for this OCD project, the answer is built into the project from the start. So the prediction targets the intervention targets the at-risk children, and the framework from work package 2 governs how targeting actually plays out in practice. So if work package 1 fails, right, we need to consider that, work package 3 falls back on the current best predictors using OCS and family history. So work package 3 is not really contingent on work package 1 succeeding, but of course we want work package 1 to succeed. But say work package 1 succeeds, then the targeting is going to be quite sharper and the trial is better powered. So anyways, this is the end of OCD section. So now let's talk about the prediction work, where I actually have some results to show. All right, second problem, predicting the trajectory of symptoms. In this case, we're using ADHD as the tool here, but you can see that it can be applied to any sort of disorder. ADHD has, if you don't know what ADHD is, I guess, it's attention deficit hyperactivity disorder. And it has one of the most variable courses of any childhood psychiatric disorder. So some children, they have symptoms that get better by adolescence. Now, some stay roughly the same, and some get worse, particularly the inattention symptoms. Now, currently, when a clinician sees a 7-year-old that has ADHD, they cannot tell the parents which group their child will end up in. Will the child get better, get worse, right? So if we could do that, or clinicians, I'm not a clinician. So if clinicians could do that, then 3 things would change. So families can properly plan, clinicians can allocate the more intensive treatments to the children who need them and lower that burden on those who are likely to improve. And it gives us stratification access for clinical trials, because currently they mix improvers and worseners and they average out their effects. So I'll walk you through some predictive modeling results that we published way back in 2021, and the design and the limitations there are going to matter for what we're doing next. So the design, we had 362 children recruited from communities around the study site. In this case, I was still at NIH, so that's Bethesda, and these were families that were enriched for ADHD. They were assessed by clinicians at study entry and then at follow-up. There was a mean follow-up of about 5 years. Okay, so in general, the kids were assessed when they were about 8 years old and then assessed again when they were 13. At each clinical visit, the symptoms, they were rated on the standard DSM count of inattention and hyperactivity. And based on this pattern across the visits, every child was placed in one of 4 trajectory groups. And you can see the trajectory groups here. We had the never affected. So these are kids who had at most 2 symptoms in either domain at every visit. So kids were never affected by ADHD. Then we have improving. So these are kids whose symptom levels dropped over follow-up. Then we had stable. So children who maintained a moderate to severe symptoms throughout the entire study. And then they had the worsening kids. So these are kids whose symptom level actually increased. And then we had predictors at baseline. And these predictors came from 3 different modalities. So we have the polygenic risk for ADHD. We have neural features, so cortical, subcortical anatomy, white matter structure. And then we also have cognition, so IQ, working memory, processing speed. And the prediction question was, from baseline features alone, can you classify a child into their eventual trajectory group? Now, for the prediction, we used a conditional random forest for 2 reasons. They handle missing data, that was the case here, and also they don't favor features with more split points the way that some other tree methods do. So we split the cohort into training and test sets, with the test set just held out for evaluation. And this is the headline result. The area under the curve was about 0.77, or above that in general, for every pairwise group comparison on the held out set. So let's break it down a little bit. Here on the top, you see the comparison of the never affected group with the other 3 clinical groups, right? You can see that the never affected versus the 3 clinical groups is usually above 0.79. If you look at the clinical group comparisons, we have worsening versus stable at 0.81, and then worsening versus improving at 0.94. But that's the easiest contrast, right? Because it makes sense, because these are the 2 phenotypically most distant groups, the kids that get worse and the kids that improve. And then we also have stable versus improving at 0.77, our lowest AUC, and that's the hardest group, right? The kids that are stable and the kids that are improving, assuming that the kids that are improving are getting closer to the stable group. Now, in terms of feature importance, we average the feature across all the different comparisons, and cognition came out on top, so cognitive features, and that was followed by polygenic risk, and then the imaging features came in third. And this ranking actually matters because cognition is the cheapest of those, right? Of those 3 modalities, the cheapest one to collect. So if a clinical version of this tool is going to exist, the cognitive features, they carry the most weight per dollar, best bang for a buck, right? Now, one thing to note here is the underlying signal, because I'm not showing here in this slide, but no single feature dominated this. So polygenic risk actually picked out the worsening group most strongly, and the cognitive deficits also flagged the worsening group more strongly. The white matter, so some of the brain features, they tracked the improving group, and the anatomy tracked the stable group. So it was quite interesting to us because the different modalities, they flagged different trajectories, which is why combining all those modalities matter. And I want to finish this particular slide being appropriately humble about this result. Because an AUC of 0.77 on a held-out set sounds a bit strong, right? But the held-out set came from the same cohort as the training set, right? So that's not really an external validation. It's the right sanity check for the data that we had, but it doesn't tell you whether this generalizes beyond the families that just walked into our clinic in Bethesda. So that brings me to the next slide. So I'm going to be quite candid about everything here because this 2021 paper, it hasn't been externally validated, not by us, not by anyone else, mostly because it's somewhat hard to get a cohort with these characteristics. So what do we know from this work? Well, an AUC of about 0.77 on the internal held-out set. Cognition and polygenic risk scores were the most informative features, and different modalities tracked different trajectories. Great. Now, what don't we know? Well, we don't know a lot, right? We don't know if it generalizes to a different recruitment site or a different country. The original cohort that we used, like I said in the beginning, it excluded children with comorbid disorders at entry. And that's a quite narrow slice of the actual ADHD population that you would see in a clinic, right? So also the cohort was predominantly white, non-Hispanic, and the mean age of the last observation of the follow-up was 13, so we couldn't really speak to adolescent or adult outcomes, and that's when the trajectory question really starts to matter. So what's next for us to do here? What's in our plan? So we're now in a position at King's to replicate this work across the cohorts that we have assembled in the last year. So the plan is, in this particular order here, is to first do a straight-up replication on at least 2 independent cohorts, ideally with different recruitment and characteristics here. And then second, do an extension to children with comorbidities, because that's what most the clinical reality is. And then third, deliberate probing of robustness across demographics and clinical heterogeneity. And fourth, extending the outcome window into adolescence and adulthood. Now, if this replication holds, then we have something. If it doesn't, then the paper still stands as a finding in its original cohort, and we'll have learned something important about which features carry through and which ones don't. All right, so let's talk about problem number 3, predicting treatment response. So right now, when a clinician prescribes for a child with ADHD, what do? They pick a first-line medication, then they watch for several weeks, and then they switch the medication if it doesn't work. The same child often goes through 2 or 3 different medications or treatments. I don't want to say it's all medication, right? It could be any treatment, before they land on something useful. And this cycle is months of suboptimal symptom control, side effects, family stress. Now, think of this. What if we could predict at baseline which intervention was most likely to help a particular child? Then we could compress the whole search space, right? So the way this gets framed in psychiatry is often as a clustering or biotyping problem. So you find subgroups of children whose biology is similar, and then you check whether those subgroups have different treatment responses. The framing is quite reasonable, but it lands in an uncomfortable methodological space because the clustering is unsupervised and the treatment response is what we actually want to predict. So the question becomes whether we should be doing supervised prediction directly or unsupervised clustering and then mapping to the response. So the work I'm going to show you now, we've done the first, but now we're moving towards the second, the unsupervised part. So here's the work that we published in this space. So Luke, Norman, and I, back in 2021, we tracked resting state functional connectivity in the brain longitudinally in a cohort of children with ADHD before and after psychostimulant treatment. And then we showed that connectivity patterns shifted with treatment in ways that mapped onto symptom change. So this was a supervised analysis with treatment response as the outcome variable. And it told us that there is signal in baseline imaging that relates to treatment response, even if the work was not framed as a deployment prediction tool. Then we also learned in this particular study that the response labels themselves are clinically very noisy, right? So think about it. So what does it mean for a child to respond to methylphenidate, right? Is it a 30% drop in symptoms? Or maybe just a clinical impression score? So the labels that we use as outcomes, they carry measurement error that limits what any model can do. And then third, the treatment response classes that we use are predefined by drug class. And that assumes the natural strata in the biology lines up with what's on the formulary. So there's not a priori reason that that should be true. So this third point is what moving us, that's the main reason here that's moving us toward this unsupervised approach. So the plan right now is to cluster on the biology first, using multimodal features, and then we validate that cluster stability across resamples and across cohorts using methods like complementary pair stability, and only then ask whether the discovery clusters predict response. Now, the clusters themselves, they are defined without reference to the outcome, and that protects against the circular reasoning of supervised biotyping. Now, again, there are no results here yet, but the methodological choices, they are quite open, and particularly around the cluster algorithms and stability assessment for this high-dimensional mixed modality data. So that's, of course, one of the places where I would like some input from the audience. And still on the discovery work, I want to point out here 3 different mega-analyses, not meta-analysis, mega-analysis that we did using a standardized pipeline across multiple cohorts. So if you're not familiar with the difference, a meta-analysis, you're using the meta-results from other studies, and a mega-analysis, we actually get the real data from all the different studies and process it the way that we want and do the entire analysis using all the data from the different cohorts. And the way we did this, because that gives us a much clearer read of effect sizes than what the published literature does, either just study by itself or using meta-analysis. And they tell us which features are worth fitting into the prediction model. I'm not going to dwell into the brain regions that are involved here, because the substantive point that I want to make for this talk is that the patterns that we found, they hold across all 3 studies, not specific anatomy. So here we have a mega-analysis in white matter. So 5 different cohorts, about 7,000 different kids, and we're looking at diffusion data, so white matters in the brain. And we use a single cleaning pipeline, and we found ADHD-specific differences, in particular the long distances of white matter tracts in the brain. So that replicated across cohorts after motion correction, which is a big thing in ADHD, but the effect sizes are small, about 0.3. And then here in the middle, we have another mega-analysis that we did, that we looked at the default mode network connectivity. And in this case, we had 1,300 ADHD cases that were matched one-to-one with controls. And we also have 10,000 strong dimensional sample. And there was a reduced anti-correlation between the default mode network and the task-positive networks. Again, there was robust to motion, comorbidity, medication, but again, effect sizes were small. And then finally, we had this paper in 2024. In this case, we did a voxel-wise analysis, looking at connectivity between the subcortex and the cortex. And we had 6 different cohorts, about 1,700 cases against about 7,000 controls. And then we had a 9,800 trait sample. So these are big numbers for psychiatry, precision psychiatry specifically, especially childhood psychiatry. And we found this atypical connectivity from subcortical seeds to the cortical regions in the default mode network and also dorsal attention networks. Again, the effect sizes were small. All right, so why am I talking about these 3 different studies? Because the pattern across all 3 of them is the same. There's real signal that replicates across cohorts, but the per feature effects are small. So this tells me 2 things. 1, these are real candidate features for prediction. And 2, individual features are not going to do much on their own. Whatever predictive power exists has to come from combining features and combining modalities. And that's exactly the rationale for the multimodal architecture in the OCD prediction work that I shared with you, and what feeds the trajectory replication plans. So these are not the features that any single one of us would have picked from the literature, but they are the features that survive when you actually pool the data. And I want to spend just 1 minute on this slide here that's also still talking about discovery, because I think it's the example I find the cleanest on how discovery and prediction loop together. Okay, so there are two different studies, two different methods, but there's one convergent story. Okay, so in this first work here, we did post-mortem work with brain tissue. So we had a paper in 2023, and we mapped the gene expression in post-mortem tissue from 60 individuals with and without ADHD in two different parts of the brain, the caudate and the ACC. Now, the headline here is that there's differential gene expression that's localized mainly to the ACC, this part here in the middle of your cortex. And the affected pathways, they were enriched for neurotransmitter genes, specifically glutamatergic genes. And we also found some correlations between ADHD transcriptome and the signatures from other neurodevelopmental disorders that suggested some sort of shared molecular substrate. Now, that post-mortem signal, that was a hypothesis to test in living children. And that's what our 2026 paper did, that was led by Marine Buisicobar. And what she did that, she used magnetic resonance spectroscopy to look for glutamate levels in the brain of living children in the scanner. And there were three groups there. There were kids with persistent ADHD, remitting ADHD, and the kids never affected. So the headline for this paper was the trajectory, right? The persistent ADHD shows an age-related increase in this medial prefrontal glutamate. And the remitting and never affected showed the expected typical age-related decrease in glutamate. The persistent ADHD group also shows an atypical relationship between glutamate concentration and the full mode network connectivity, which links this finding directly back to that DMN-MEG analysis that I showed in the previous slide. Now, what makes this relevant to the prediction modeling talk is because you have to notice the structure of this finding, right? It's not children with ADHD have more glutamate. It is children whose ADHD will persist have a different level of glutamate trajectory. And that's a trajectory result, exactly the kind of feature that should feed into the trajectory prediction work that I described earlier in the 2021 paper. So when we replicate and extend that 2021 work, glutamate trajectory becomes a candidate feature and one that we have biological reason to take seriously thanks to the post-mortem work evidence. Now, I want to be clear here that I'm not claiming that we have used glutamate as a predictor. We haven't. But that's the kind of feature that earns its place in the next round of models because it has converging evidence, right, from two methodologically independent approaches, and because the finding itself is structured the right way for the prediction question. This is what I mean when I say discovery feeds the prediction work. All right. And I'm going to close with the Pears Maudsley Centre that I have mentioned the data problem repeatedly during this talk, right? And the cohorts that produce the most published psychiatric prediction models, they systematically exclude the children who most need help. Now, the severely affected children, like I said, they move too much in the scanner and they get excluded from the analysis because you cannot just be decoding motion, right? Now, children with comorbidities, they are also excluded by design from the clinical research cohorts because you want a very clean ADHD signal. And demographically, the children, the children that are highly diverse, they are underrepresented because of where the recruitment for these studies, where they usually happen. Now, each one of these exclusions is maybe individually defensible at the level of a single study, but when you put all these data sets together, right, cumulatively, They mean that our models are calibrated on the easier patients. Now, what the Pears Maudsley Centre is being built specifically to address that. And beyond just collecting better data, what I really want the center to be is a hub for the kind of multi-cohort precision psychiatry work that this talk has been all about. So that means a hub for data, but it also means a hub for the analytic methodology that lets you actually use multi-cohort data well. So that includes cohort harmonization, cross-cohort validation strategies, fairness assessment across demographic groups, and all the methods for handling the structural missiness that comes from combining cohort that's been collected on different protocols across many decades, right? So the center is positioned to develop and disseminate all those methods and not just feed its own pipeline. And if precision psychiatry is going to scale beyond a handful of well-funded labs, methodological infrastructure has to be part of the deliverable. So let me show you briefly what the center is and why I think it matters. It's just one slide. So there are 4 things that the center does, and I think make it more than just another well-equipped imaging facility. So first, severely affected children. So the center includes movement-tolerant tools, including the OPM-MEG that I'm heading, which lets us record neural data from children who would never make it through a conventional MRI session. That single change opens up a population that has been missing from the field's cohorts. Second, demographic diversity. The catchment area in South London is genuinely diverse, and the recruitment design is deliberate about it, instead of just relying on whoever happens to walk through the door. This is the harder problem to solve, and I really don't want to really overclaim it, but the architecture of the center is built to make this tractable instead of just ignored. And then third is the real-world clinical phenotype. The recruitment criteria do not exclude comorbidity. So children come in with whatever their actual clinical picture is, including the cross-cutting symptoms that don't fit cleanly into the DSM categories. A child might have several different symptoms of several different disorders. And honestly, that's uncomfortable for some kinds of clinical research, but it's exactly what we need for prediction modeling that has to work in the clinics. And then fourth is the deep longitudinal phenotyping. We're talking about multimodal imaging, genomics, cognition, behavior, clinical trajectory, all of that collected on the same children repeated across development. The cohort that comes out of this will be exactly the kind that the trajectory and the treatment response questions need. All right, so let me close this with 3 asks. First, methodological feedback on the design choices that I flagged throughout this talk. And you know these problems better than I do, and the choices that we make in the next few months will shape what the lab produces for the next several years. Then second, we're definitely looking for collaborators for cross-cohort validation. So if you work with a cohort that could externally test the trajectory model or that overlaps with our OCD outcome, I'd love to talk. Or if you have students that need data to play with and would like to collaborate with the PANDA lab, yeah, just ping me. Now, third, the lab is hiring. So if you know people that are working on prediction in adjacent settings who might want to bring those skills to pediatric psychiatry, please send them my way. So thank you for this time, and happy to take questions.
Thank you so much, Gustavo, for the very, very clear, very informative, and very exciting as well. I really like the way that you are closing your talk of giving all those opportunities, right, to collaborate and making this really interactive. And so, yes, I think that now is the moment for the audience. I think that there is a first question by Saskia. Yes, please go, Saskia. Thank you.
Hi, thank you. That was super interesting. So I was thinking about your work packages 2 and 3 in your first welcome project, and one of the things I'm oh sorry, do you want to just mute because I know you're in public space? Thank you. Yeah, so on work packages 2 and 3, I'm going to put in the chat a paper that I wrote with Mike Inouye about clinical implementation of polygenic risk scores in practice. And so the reason for that is we have been, well, over the years, I was actually previously at NIH, at NHGRI, and over the years, I've done research on the sort of clinical applications of interventions that incorporate genomics, including polygenic risk scores. So I thought this might be helpful for you as you think about how you're translating into going towards work packages 2 and 3 and how you involve patients and the public in the design of that. And I would say also my suggestion methodologically here, building on what we've put in that paper, is that in addition to the very important involvement of people with relevant lived experiences in the co-design of what you're doing, I would also look to the existing evidence and best practices, of which there is quite a lot in the genomic space, but means much, much more, and underlying behavioral and other types of theory as well. And then I will also, so that was just one point thing on the methodological side. I also wanted to say that from the data side, we work closely with the NHS Digitrials team and the central NHS data team at what's currently NHS England. And so there may be some interesting things to look at in the National Secure Data Environment, SDE, through that resource. And again, I could connect you with the relevant team if you're not already connected with them through that. It's a really useful way into looking at national data across everybody, across the whole nation. And then related to that, if you're looking to think about how you recruit cohorts or identify people from across the whole of England to invite into specific cohort studies at the new centre, that could also be, there's also a recruitment service arm of that. Again, work very closely with them, and that might be something that might be useful to you going forward. So I just wanted to mention those things. Oh, and the last thing was, again, you're probably already linked into this, but we're also starting to work quite closely now with the South East England Genomics Medicine Service. So as you're probably aware, the Genomics Medicine Services, as part of the new 10-year plan, have been, sorry, I know I'm going on, but I will stop after this point. The Genomics Medicine Services are now, in addition to their existing 2 pillars of working on rare disease and cancer, they're expanding now to a 3rd pillar across the whole nation to population health. And it's very likely that is going to include pharmacogenetics, so obviously the genetic responses or genetic influences on drug response. And I think as that program gets going and we work across partners on service delivery, but also the generation of evidence and finding the right use cases for both research and care with those clinical partners, I think that could be also a useful place for you to make some new connections if you're not already connected there. So very happy to pick any of that up with you afterwards by email or anything else.
Well, thank you. This is extremely helpful, and thanks for the paper. I'll definitely follow up with that. I was at NHGRI as well, so I wonder if we overlap there. But I think this is very important, and I'm certainly going to read the paper and follow up on these connections, because we definitely need to contact them to make this work even bigger. But also, because you mentioned, so we do have people working in work package 2 in terms of the interaction with PPIE, but it's very important, especially if you're talking about PRS, right? How do you communicate that the risk is mostly already in your genes, right? What does a person do with that, right? How do you communicate that? So perfect. Yeah, I'm very happy that you share these sources, and yeah, I'm certainly going to follow up with that if that's okay.
Lovely, thank you. Great.
Thank you. Great stuff, Kev. We have more people raising their hands. I think that now it's the turn of Liv.
Yeah, hi. Firstly, thank you so much for this talk. This was really fascinating. And I really want to add to exactly what Saskia was just saying and what your comment back to that was. So I know that when looking at prediction and children especially, there's always that issue of whether to flag something or not. So with this kind of research, and say you did manage to actually generate an algorithm that can predict ADHD or OCD or any kind of mental health that comes with a stigma, how would you go about calculating where that threshold is that would be worth warning the parent due to that potential stigma and repercussion of just flagging it, even though it might not even end up presenting?
No, that's a very important question, and that's why we count with the work with people, the interactions with people with lived experience, clinicians, families, because that's as important as the prediction tool itself, right? Because this is all part of the same work. So we are very aware that that's not a question that we can answer by ourselves, and we should not answer by ourselves. So in terms of thresholds that we define what's worth communicating, how we communicate. So that's all work that needs to be done together with the community. So yes.
Right. Yeah. It's very challenging. I'm looking at the same thing, but with depression and.
Oh
Yeah.
yes, I hear you.
I might just
Yes.
shoot you up an email just to discuss that further, if that's okay.
Of course, yeah.
Yeah.
Happy to.
Thank you. All right. Thank you so much.
Can I make a quick point on that? Because probably you already know, Gustavo, but you know that we have these different guidances on how to communicate this type of information, and also related with the responsible prediction modeling, responsible AI. And there are two particular guidances on that, one from UNICEF and the other from the Convention on Rights of Children. And both of them cover, particularly for children, how prediction models should be presented, how the information should be communicated to patients, and so on. I think that can be helpful in your case.
Oh, for
I'd be
sure,
happy
yes. That would be,
to get in touch about it if you would like to.
Perfect. That would be very helpful. Thank you so much, Raquel.
Lovely. I think that now it's Daniel. We have 6 minutes yet, so
yeah.
yes, Daniel.
Yes, thanks.
Just don't make it too hard, Daniel, because I don't want to end up the talk with a question that I cannot answer.
No, it was a really great talk, and I think you did. I covered everything very well, and I think you tried the best to answer your questions. I can't really say what's better because there are always the same problems, but just to cover one problem, perhaps, is the prevalence of the first study is 2%. So if you have got a prediction modeling, you need a sensitivity of 95, 99%, that you don't really have most of them as false positive in your model. So I don't know how it will work in practice because, it will be, that's the problem. The second is also with question with prevalence. I like that you want to do this leave one study site out approach, but why would you not do it with all 9 data sets? Because you've got a larger sample size, especially with this low prevalence, you still have got a lot of information about generalizability.
No, yeah, these are 2 points that we have struggled a lot with. I also don't have a real answer. So in terms of the imbalance of the classes, right? Of course, we're going to be using for evaluation everything that can, all methods that can account for that. So we're not fooled by the class imbalance. But yes, it's we have to run the data to see what we can get, and we're hoping for the best here. And in terms of the leave one cohort out, that was a strategy that we started with, and we like that. But I've been pushing back with our collaborators because there might be information in the cohort itself, knowing what the cohort comes from. And my point there is mostly because, say we come up with a great tool, and we talk to community, we know how to communicate properly the results. Then if this is to be implemented in a new site, I have a hard time thinking that we wouldn't retrain the tool with data from that site, because I imagine that the predictions would be better, more refined with data from that site. So that's an argument for putting all cohorts there at the same time. So my gut feeling right now is that we will do it both ways. Of course, report both, but that's my current feeling. I don't know if you have any insights other than that.
No, I think it's, as you said, a difficult problem. I'm sure there are different countries. I don't know where the cohorts are from anymore, but if you have got different countries, of course, you might want a very different cohort where you do which you use as external data.
Yeah, there's that, but also you mentioned the different counters, right? But like the more you start working with the data, you realize that, well, this cohort, you have to do the analysis just in their theory. You cannot bring the data here, you cannot bring the data there. So that's actually going to force our hand on how much you can put the data together, what can be replication, what can be discovery. So those are things that we're only discovering now as we start to do the work.
Yeah. I mean, perhaps we can discuss it more in the prediction modeling seminar, because I think we have got not much time left and other people might want to ask questions, but it's a really great study, and well, the Wellcome funded it, so they also liked it.
And we're happy with it too. And actually, thank you again to you and Raquel, because you helped us prepare for the interview. So yeah.
It was great that you could get it. So our pleasure. Okay, so it's 2 minutes to go. I don't think that there are more comments or questions from the audience. Anyone else would like to add anything very quickly? Okay, if not, we will leave it here. So Saskia is adding some information about the BRC Informatics Forum that is also working. You can read it in the chat, Gustavo. That could be of interest. And we're going to leave it here, but not before saying thank you, a big thank you to Gustavo again. It has been a pleasure. So thank you so much for presenting your projects, and we really hope that we can collaborate in the future and that you are very successful in your project. So thanks, and thanks everyone. The rest of the audience for attending. Hi everyone, and bye everyone. And also would like to tell you that in May, on 27th of May, we will have our next machine learning and prediction modeling seminar. This is going to be by Professor Marta Maudsley. He's a professor on responsible AI in mental health care. So this could be of interest of anyone here as well. And she's going to be talking, the title is going to be AI and Mental Health Equity, Reimagining the Future of Clinical Decision Support. This is 27th May, 3 to 4 p.m. So everyone welcome and have a lovely time until there. And also today, enjoy your evening. So thanks everyone. See you soon. Bye.
Thank you. Bye-bye.
Bye-bye.